All right, so we've seen how you go from alpha-ketoglutarate to glutamate and glutamine. A couple of words for proline and arginine, and from now on, the enzymes really become. There is a long series of reactions. We're going to try to go through them, not in detail, but focusing on some major themes, hopefully, and see what we learn. So, all right, let's see proline. All right, so start with proline. So this is a glutamate, and this carboxyl group will get activated. It's going to be phosphorylated. That's where you spend ATP, and then you reduce this carboxyl carbon to the aldehyde. So you use reducing power, and then spontaneously, once you have this product, will cyclize to give you this intermediate, which then you have to reduce again, so you don't have this double bond, and you get proline. Do we need to remember all these things? No. So what's the point of this? Well, remember, this is anabolism here, right? So we start with something simple, and we want to make something a little more complex, although not as complex as a whole protein, just the building block here. But the whole idea from our metabolism scheme was that whatever ATP you made from glycolysis or oxidative phosphorylation, and all the reducing power you got, let's say, from the pentose phosphate pathway, this is where you put it to use. Okay, so this is going to be the theme. Okay, this is where you spend your energy. You're reducing power. You have important building blocks. Let's say TCA cycle, alpha-ketoglutarate gave you glutamate, and this is where it all comes together. But it comes together in so many different flavors that appears complex. And to be honest with you, unless you actually work on these particular reactions, it's impossible to remember all of them. So let's stick to the principles here that we're showing. How about glutamate to arginine? Here we're going to take a little detour because there is a special aspect to it for human physiology. But again, we see the same theme. So it's very similar. It starts out from glutamate, and it's extremely similar to what we saw with proline, but with some twist. Instead of phosphorylation with ATP, here we have acetylation of the carboxyl group. Why is that? All right. So you keep going with the same reactions, and we have an acetylation here. First, sorry, ATP comes next, but before you do the ATP, this part has to be it has to be acetylated. The amino group has to be acetylated, and then the ATP comes in. So why? Let's go back here. Here we just acetylated straight up. Okay, so why didn't we care about modifying this amino group on glutamate? In fact, that was great because spontaneously, if this amino group is there, it will close up to give us proline eventually, right? But proline is the only cyclized amino acid, no other amino acids like this. So it is not great to have a free amino group floating around here. And that is the reason why you block it effectively. You block the amino group with acetylation, with acetyl-CoA, and then you start on the usual reactions, okay? You activate this carboxyl group, and then you reduce it, you make the semialdehyde, and then you bring in more glutamate, and you make N-acetyl L-ornithine. If you didn't protect this amino group, then it would cyclize automatically, so you wouldn't go to these next steps, right? So again, same principles, and you see how nature finds a way to just build on them and make everything modular. So we haven't seen anything about arginine, but this is how you get to an amino acid that's natural. It's found in our cells, but it is not found in proteins. This is called ornithine, right? Oh, sorry, the last step, you remove the acetyl group. Okay. So this is L-ornithine, and from L-ornithine, you can get to arginine biosynthesis. You can get also back to proline biosynthesis and some other more specialized natural products. So for arginine, we have to deal with the urea cycle. So this is, we're going to take a little detour. If you look at ornithine and how arginine looks like, there is this extra group, okay? There's carbon and two more amines. So this is a heavily aminated, it's the most aminated amino acid, right? So there is a lot of nitrogen on arginine. So somehow we have to go from ornithine, which we saw in this scheme how we got it, which is fairly straightforward, very similar to proline. but we have to add to it 1 extra carbon and 2 nitrogens. And this is what we have discussed, the urea cycle. It's an extremely important sequence of reactions, again, for a variety of reasons. The most important of which is this is how we excrete excess ammonia in our urine, right? So we excrete in the form of urea, which is generated in the urea cycle, okay? But before we get there, let's see how we start everything. So we have to go back to the basics in the mitochondrion, and we have a carbamoyl phosphate synthase, this enzyme here that generates carbamoyl phosphate. And that is made out of CO2 and ammonia. So there is quite a bit of CO2 and ammonia forming in various reactions, and especially in the cell too, in mitochondria. And ornithine will come in and combine with carbamoyl phosphate. We'll describe how that reaction happens with carbamoyl phosphate in a minute, carbamoyl phosphate synthase, and generate citrulline. Citrulline is a bigger version of ornithine. There are special shuttles that bring ornithine in to the mitochondria and take citrulline out. Once citrulline is out, it combines with aspartate, and of course ATP is consumed at various steps here, including in this one, and makes this dipeptide, arginine and succinate. And then from that you get arginine. So this is actually the actual biosynthesis of arginine is all the way from ornithine combined with carbamoyl phosphate, going through all these steps, and finally you get arginine. If you were wondering how we get arginine, you release fumarate also at the same step. And what's important here is that arginine, now you can hydrolyze this part, okay? The 1 carbon and 2 amino groups from arginine, regenerate ornithine, okay? But also generate urea, okay? Which now it can be excreted. And this is really how our cells manage to sequester excess nitrogen in the form of ammonia and CO2 also, and excrete it in the environment. Ammonia is a poison. Okay, if you build up too much of that of ammonia, especially in the brain, you have some very weird hallucinations apparently, and then it's lethal within minutes. Actually. So maintaining proper function of the urea cycle is absolutely essential for ammonia for ammonia homeostasis, which we don't want to build up excess ammonia. As I said, this sequence of reactions leads to arginine, so some of it, if you need arginine, will be taken out of here and be incorporated to proteins. But arginine in this case is an intermediate in this cycle, but the job as it is drawn is to really generate urea out of these products in the mitochondria. So there is a enzyme that makes carbamoyl phosphate. There are 2 versions, one in the mitochondria, one in the cytoplasm actually. So the mitochondrial one is version 1. It's super simple. The products, the reactants are extremely basic. Where is the ammonia coming from? Some of it is coming actually from this reaction, as it turns out, glutamate dehydrogenase. When you go from glutamate to alpha-ketoglutarate, you generate quite a bit of ammonia. And in some settings, that contributes the most. Not always, but it does. Oh, why also in the brain is especially important not to accumulate too much. So glutamate is, in addition to all these metabolic roles, it's a neurotransmitter. So there is a ton of glutamate produced in our nerve cells. So if you want to remove that, of course, you're going to go through this reaction, generate a lot of ammonia, and then the urea cycle has to kick in. The mechanism, we've seen how you need to activate carbonate. Okay, we've seen this before. And once you activate carbonate, now it can be attacked by ammonia. And then the second ATP comes on to give you carbamoyl phosphate. Right? So fairly straightforward. And we've seen already half of this reaction in previous lectures. And that closes up the urea cycle. A lot happens, although as I said, happens a lot it happens in all cells, but the liver and kidney are the primary movers in this case. We do not secrete ammonia into our urine. Our we secrete mostly urea. Okay. Now, I had this question, believe it or not, in class. How come you go to public bathrooms and you smell ammonia? I mean, you take an ammonia-based cleaner and you know how ammonia smells, and you go to a public bathroom sometimes when it hasn't been cleaned and you smell ammonia. So how is that possible? Once you urinate in the environment, there are bacteria that have ureases, and they take the urea that animals secrete, let's say, and cleave it, giving rise to ammonia. Okay. All right, so now let's move on to additional amino acids. And again, to avoid the super confusion, I will try to stay with basic principles. So lysine biosynthesis also starts with alpha-ketoglutarate, not glutamate. And the first part is something we have seen before. It's a +1 version of the TCA cycle. And what do I mean by that? Well, we have alpha-ketoglutarate, and you get acetyl-CoA, and you get homocitrate. So that, if you go back to the TCA cycle, alpha-ketoglutarate has 5 carbons, right? But the way it works with citrate synthase is to get oxaloacetate, which has 4 carbons, combine it with coenzyme, with acetyl-CoA, which has 2 carbons, and get 2 plus 4, 6. Here, we start with 5 plus 2, and we get 7. And that 7-carbon compound is called homocitrate. It's a different enzyme than citrate synthase, but clearly it's an enzyme that evolved with tiny modifications, maybe a bigger active site, to accommodate the extra carbon on the substrate. So it can pick up a bigger substrate. But the same mechanism is used, exactly the same reaction. And the rest of the reactions are, again, identical to the reactions we see in the TCA cycle. Instead of ending, if you start with oxaloacetate, you're going to get to alpha-ketoglutarate. Here we get to alpha-ketoadipate, which everything here is 1 carbon bigger than the canonical TCA cycle reactions from oxaloacetate to alpha-ketoglutarate. So it's actually kind of cool. This is the canonical TCA cycle, right? This is the 5-carbon unit, and we get to the 4-carbon unit, oxaloacetate, and we get to the 5-carbon unit, alpha-ketoglutarate. Now you take the exact same reactions, you modify the enzymes a little bit so they can accommodate, substrates that are 1 carbon bigger. So you make these reactions that we discussed earlier. That's the alpha-ketoadipate. That's alpha-ketoglutarate now as a substrate, but everything else is exactly the same, which is a beautiful example, again, how nature is modular, biology is modular. You build a fundamental module, you know that these reactions work. They're the best way to get you from this type of compound to this type of compound. If you want to generate just a little bit more of a variety with an extra carbon, you just make the enzymes a little bigger, and everything else is all set. And once you do this, that's alpha-ketoadipate that happens in the mitochondria in the modified TCA-like reactions, and you bring it out and you follow it up with extra reactions to get what you want. And at every step of the way, you use reducing power, and you get what you want. And that closes this sequence of events. Okay, how we go from alpha-ketoglutarate to all these different amino acids, the 5-carbon amino acids, so to speak.